We're ready to move on to our next speaker, which is Emmy Felker-Quinn. She's going to be talking about interpreting terrestrial sulfur critical loads to protect national parks. So please feel free to share your screen, Emmy.
All right, well, thanks for joining us today, everyone. My name is Emmy Felker-Quinn, and I'm in the Air Resources Division at the National Park Service. Today, I'm going to be talking to you about interpreting terrestrial sulfur critical loads to protect national parks.
Emmy, you're not in presentation mode if you intend to be.
I don't. I'm having some trouble with presentation mode, so I'm gonna stay in this mode.
Okay.
There we go. Thank you, Linda. So today I'm going to start by just stepping back and telling you about the data set I'm using. Some of you know it as the Horn critical loads. It's this enormous analysis of 94 common tree species from the Forest Service Inventory and Analysis Program, put into models of temperature, precipitation, and total nitrogen and total sulfur deposition averaged over the years 2000 to 2012 to, through model selection, generate a possible set of 4 relationships between either nitrogen or sulfur deposition and tree growth and tree survival for individual species in the continuous United States. I'm focusing on the sulfur critical loads. The models in the model selection in this analysis reflect acidification impacts of sulfur on tree growth and survival, which is to say that models used for sulfur generally show decreasing growth or decreasing survival over sulfur deposition. So by the classical definition of a critical load as the amount of deposition that a ecosystem or organism can tolerate before we see negative effects, the critical load in these relationships is set at the maximum growth or survival rate, which also means that it's set at the minimum sulfur deposition experienced by a tree species across its spatial range in the US. I should mention that as the National Park Service, we're required by the Organic Act to preserve parks unimpaired, and so we're pretty okay with the idea of a really low critical load. The advantage of these continuous models is that it also allows the calculation of exceedance when deposition exceeds the critical load, and to tie it to a specific amount of tree growth or tree survival decline. So I'm going to be showing figures based off of these curves throughout. And basically you can see as the color gets more saturated and darker, either darker red or darker blue, deposition levels would predict 1, 2, 5, or 10% decreases in growth or survival. Some of the tree critical loads calculated within this data set are low. The critical load can be as low here for Canyon live oak as about 0.5 kilogram of sulfur. And importantly, the exceedance decline points are also very low. So based on sulfur deposition, you would expect, you would predict that Canyon live oak could be at a 10%, more than 10% growth decline and 2% decline in survival rate at deposition level of 1 kilogram. And within the set of 40 trees with any sulfur critical loads and 51 trees with any sulfur survival critical loads, there are 10 species with the sulfur growth critical load below 1 and 9 species with a sulfur survival critical load below 1. And each of those 14 species, we would predict declines at sulfur deposition rates less than 1 kilogram. And so the question is, should we apply these critical loads broadly across the Park Service? To sort of address this, we first asked the question of which National Park Service units have sulfur critical loads. We did this work in contract with Kevin Horn and Quinn Thomas, who authored the original paper. And for this, we use the NP species database, which is basically a species list compiled by participating parks. And we joined our park-specific species lists with the critical load values. And these maps on this slide show where the parks are that have any sulfur critical loads, below 1 or above 1. And you can see most of the tree species within the parks that have sulfur critical loads are in eastern parks. It's up around 30 to 40 species in eastern parks, and in western parks, it's more like 14 parks that have up to 14 species that would have critical loads. So this reflects both tree diversity and how the FIA collects data. So it's not a surprise. When we look at the subset of sensitive trees, the trees that have sulfur critical loads below 1 kilogram of total sulfur, we see that they are concentrated mostly in the West. We have a few broadly distributed national tree species that are also in the East, but it's mostly our Western species. Again, it's not too much of a surprise when you start with these kind of model that the trees that occur in these relatively pristine Western parks where sulfur deposition is so much lower than in the East, that these are where those critical loads end up. But the corollary question is, are there parks where we only have sulfur critical loads below 1? And so this map shows the subset of parks in the West that only contain sensitive species with critical loads below 1. So there are parks where the only growth critical loads are below 1 kilogram of sulfur in red. There are 22 of those parks. There are 39 parks where any of the sulfur critical loads for both growth and survival are below 1. And there are 3 parks that only have survival critical loads below 1. Just to sort of magnify why this is an important subset of parks, 19 of these 64 parks are Class 1 areas, so are afforded special protection under the Clean Air Act. So then the question is, what are the predicted consequences of current deposition in this subset of parks where we only have sulfur critical loads for these sensitive species? And so to sort of address this, I'm going to join the most recent 3-year TDEP total sulfur average from 2016 to 2018 to species presence and absence of these sensitive species in these parks. And when I do that, you can see that there are declines predicted under current sulfur deposition in this subset of parks. So for this graph, a single column here represents all the instances of a single species across this whole subset of parks. And you can kind of see that for many of these species in this western range of parks, we would predict more than 10% reduction in growth rate based on these relationships and current deposition rates. There's a similar but not quite as dramatic pattern for sulfur effects on tree survival, where we have declines predicted in many of these parks. So to summarize, we have 39 parks that only contain tree species with sulfur critical loads below 1 kilogram of sulfur per hectare per year. And based on these critical load relationships and the exceedances, we would predict substantial declines for these tree species within these parks. And that's a pretty extraordinary result because it doesn't necessarily correspond with what we think we know about these parks. And so it offers an interesting opportunity to sort of revisit what we know and compare the FIA data, which is collected in forests outside of national parks, to some of the forest growth and survival data collected by the Park Service. but it still leaves the question of how to summarize these critical loads for parks. And so I want to focus in on Bryce Canyon National Park, which is one of these Class 1 areas pictured here. And at Bryce Canyon, it's one of those parks that only has sulfur critical loads below 1 kilogram for both growth and survival. And so if we're going to apply the horn critical loads directly and quantitatively to the park, we would say that this park has 4 trees with sulfur growth critical loads and 5 trees with sulfur survival critical loads. And based on current deposition rates in the park, we would predict declines in both growth and survival at current deposition. We're also considering applying the sensitive sulfur data more qualitatively. So, for example, the Park Service uses lists of ozone-sensitive species and lists of acid-sensitive species to identify species that we know are at risk, but that for which we don't have quantitative dose response or critical load values. And so what we could say at Bryce Canyon National Park is that we don't have quantitative sulfur critical loads, but we do know that there are these 4 tree species likely to be sensitive to sulfur effects on their growth, and 5 tree species sensitive to sulfur effects on their survival. And so we're kind of weighing these options of quantitatively applying the critical loads, or of using the information from this analysis to say that we have identified sensitive species qualitatively in the Western parks. And so with that, I would like to acknowledge Kevin Horn and Quinn Thomas, who did the original work and with whom we're co-authoring a report on applying the nitrogen and sulfur critical loads within the national parks. And I'd like to acknowledge Justin Coughlin, Chris Clark, and Jen Phelan, who are leading efforts to push the analysis of the tree critical loads and the application of the tree critical loads further. And with that, I thank you, and I'll take any questions.
Oh, thank you, Emmy. Are there any questions? I do not see any in the chat box, but if you stop sharing your screen, we'll be able to see folks, and we may be able to get some questions directly from them. We have quite a bit of time left, so feel free to ask a question. You can just unmute yourself.
I have another question. Hi, Emmy. It's Jill. So I was wondering, like, in light of like Mark Fenn's recent work showing that, you know, that there actually are some benefits to sulfur deposition, and the fact that the horn curves, like, they immediately start, you know, they don't allow for that increase in growth or survival like the nitrogen curves do. Is anyone working on like reassessing these sulfur deposition curves to see, you know, because sulfur is an essential nutrient, and you know, and we know that some of those low deposition rates don't necessarily align with, you know, with soils, like what this, you know, like soil science would tell us and processes. So
yeah, that was an interesting result in the California FIA data set, that they found that growth correlated positively with both nitrogen and sulfur deposition. And we're working with Chris Clark and Jen Phelan. Jen Phelan and RT are leading an effort to redo the tree critical load analysis, split region by region. And so I think we're discussing whether to include the sulfur parameter as not just in that decreasing relationship, but possibly in the hump-shaped relationship that we do for nitrogen. Yeah, that's been an interesting result that we're trying to figure out how to incorporate.
Yeah, and Emmy, I can follow up a little bit if you want.
Mm-hmm.
Yeah, so we are looking into that, like Emmy said. I've been emailing back and forth with Mark a little bit because I'm trying to get a better understanding of which correlations they evaluated beforehand. I could not find them in the paper or the supplemental. So I'm not sure if they had that cross check. So I think the study is great, and I think it's really an important contribution to the literature. But from our purposes, I really want to get to the bottom of how much checking ahead of time on independence was done before those curves were generated. Same problem we have, that Linda Geiser has brought up in the past for the horn curves, that there are high correlations for some species. And so those curves should be used with caution. And I couldn't find that anywhere in the paper.
We have about 3 minutes left. Anybody else with a question or comment?
Well, I have one in the chat, if I can read it.
How did I not see that?
I should probably know this, but I don't. Sorry. I mean, did you guys do the same cross check that we were just talking about, like which species, if they had a really high NS correlation, either you didn't use it or you flagged it or something like that?
Can you hear me?
Yep.
Great. Okay. So I'm showing the figure from Kevin's paper. We didn't flag any of the species with the high VIFs, and basically what Kevin published in his paper was just to flag that sulfur deposition in the western part of his analysis is highly correlated with nitrogen, with temperature, and with precipitation. And so I think that is another way that we could sort of decide how to apply these critical loads from this data set is to look at the VIF and use certainty based on that.
Thanks. Yep.
Okay. Well, I think that might be the total of the questions for now. And everyone, please feel free to follow up with Emmy afterwards. And we can go on to our next speaker, which is Nicholas Russell. He'll be talking about exceedances of critical loads for herbaceous species in national parks, species at risk, local responses, and regional trends. You there, Nick? Just go ahead and share your screen then. The floor is yours.
Great. Thanks, Linda. So I'm Nick Russell. I'm an intern with the National Park Service Air Resources Division and a grad student at UC Denver with the Department of Geography and Environmental Sciences. So today I'm going to be talking about some work I've been doing looking at applying some critical loads for herbaceous species to national parks. So first, a bit of background. As was mentioned previously, critical loads are sort of that safety point below which you're not going to see negative impacts from, in this case, nitrogen deposition. Recent research in critical loads has mostly been for broad spatial regions or groups of species, but there has been some growing research on individual species, as Emmy just presented on. But some of the limitations with just having broad spatial critical loads or groups of species is some of that variation of an individual species response can be lost, because responses to deposition can differ across habitats due to environmental factors like precipitation and temperature and soil or other deposition characteristics like sulfur. And so the National Park Service uses critical loads to help preserve and protect natural resources. And species-specific critical loads could really help identify most at-risk species within parks and across parks. And it would help allow the prioritization of species and management needs in national parks. So as I mentioned, there has been some work on creating loads for individual species. In 2019, Clark et al. Published a paper that created critical loads for a set of herbaceous species. And they did this using probability of occurrence models, which you can see there on the left. And those were based on nitrogen and sulfur deposition data, soil pH, and temperature and precipitation data. And they estimated the critical loads at the level of nitrogen deposition above which the probability of occurrence would decline. So just taking a closer look at the input data that they use, because this study is sort of what we're basing our application of the critical loads on. They used roughly 15,000 vegetation plots, which are mapped on the left there. And from those plots, they selected 348 species that had enough data to create the probability of occurrence models, and were able to create critical loads for roughly 200 of those species. And as I mentioned, they used nitrogen and sulfur deposition data, and they used long-term looking at from 1985 to 2011. And they also used 30-year normal temperature and precipitation data, along with soil pH from the USDA. This map just shows, again, that input data from the Clark et al. Paper and the national park boundaries in green. And you can see that there is some overlap, but especially in the west and northwest, there's not a lot of overlap between the plots and the national parks. So that raises the question of how we can confidently apply these critical loads to areas outside of that were used in the original models. And that's sort of the main research question that is being asked here is, can these existing herbaceous species critical loads be applied to national parks? And we assess this in two different ways. First, sort of just by directly applying the data. So we basically just took the existing critical loads and applied them to parks to see what they predicted for species response. And then we took a bit of a deeper dive into the data and the models by trying to see if we could input park-specific data into those models to evaluate expected species response. So for the direct application of the critical loads, we use a standardized species database, NP species, that Emmy mentioned, which tells you whether or not a species occurs in a national park. So we don't know the specific location of that species, like a plot, but we know whether or not it occurs in a park. So we collected that data for all of the species, the 348 species from the Clark et al. Analysis, and then we're able to match the critical loads to that park species data. And then from there, we evaluated critical loads against total nitrogen deposition from 2016 to 2018, or the average from those years. And then moving on to our deeper dive into the data and the models. So first, what we did was we selected 9 case study parks, which you can see on map on the upper right. And then within those parks, we used vegetation inventory data to locate species within those parks. So that map on the lower right shows some of the plot data that we have that's for Great Smoky Mountains, but we had that data for each of our case study parks. And each of those plots has at least one of the 348 species from the Clark et al. Analysis. And since we have this specific data in these parks, we can then join the specific environmental data at those locations as well. And after that, we checked if the input data that we're using that's getting joined to those plots falls within the same range as the data used in the Clark et al. Models. And from there, we then input our data into the models to calculate probability of occurrence. And again, this just shows the input data versus from the Clark et al. Analysis versus the plot data that we have, and that's in green in our case study parks. And this, one of the main differences that we had in our data versus what was used in the original models by Clark et al. Was instead of using long-term nitrogen and sulfur deposition data, we're just looking at the average from 2016 to 2018. But the rest of the data was the same. We also used 30-year normals for precipitation and temperature, and also soil pH data. So looking at some of the results from our direct application of the critical loads, we found that median nitrogen critical loads across national parks range from about 6 to 19 kilograms per hectare per year. And that of the roughly 244 parks that we included in our analysis, 220 of them had at least one of the species with deposition and exceedance of its critical load. And you can see on this map here that the majority of the exceedances are clustered in the mid-Atlantic and eastern regions and the upper Midwest. And you can also see that here in the southwest, we see a cluster of parks that have low exceedances or no exceedances. And that's partly due, as I showed in those maps from the input data, that data set is fairly East US heavy. There aren't a lot of plots from those areas, and the species included in the analysis might not be as common in the Southwest either. So we have a bit of a data gap in that region. And then we found that an average of 17 species had deposition in exceedance of their critical load per park. And I think this direct application of the critical loads, while it's really simple and basic, I think it does provide us with some really interesting and useful results. That table up there on the top left, that shows the top 5 most common, or the parks with the most species with deposition and exceedance of their critical loads. And the column on the very right shows the total number of species from the Clark et al. Analysis that we found in those parks. So it can give you a pretty good view of an individual park and how that park is dealing with nitrogen deposition for a particular group of species. But it can also give you species-specific information. And that table there at the bottom shows the species and the number of times they occur in a park where their critical load is exceeded by nitrogen deposition. So you can see which species are most sensitive or most often in exceedance across national parks as well. And moving on to our deeper dive into the models, this is just a quick summary of our data from our case study parks. This table on the left shows the number of vegetation plots we have for each of our case study parks, followed by the number of total species found in those plots. And then finally, the number of species from the Clark et al. Analysis that we've located in those parks. And this again highlights the sort of data gap we have in the Southwest, because you can see that Joshua Tree only has 5 of those species in it. And then the table on the right, that just shows the most common species across all of our case study parks. So the next thing we did was compare our input data that we're using in the models versus what was originally used in the models. So here we selected 3 of fairly common species, both in our data and the original plots from the Clark et al. Analysis, and compared the range of the input data. So you can see that for nitrogen deposition and soil pH, our data does fall within the range of the original data. However, the nitrogen deposition data we have is a much more narrow gradient than the original data. Ours only goes up to around 7 compared to 15 to 19. And then for temperature and precipitation, we're fairly close to the range, but we're pushing a lower bounds for temperature and the upward bounds for precipitation, at least for these 3 species. So next, and applied those to the probability of occurrence models provided by Clark et al. And compared our results to the original results from that paper. And you can see here for Equisetum or Vence that our results follow pretty closely to the original response of the species from the Clark et al. Paper. It's following that decline curve there. And then here on the right for Poa pratensis, this is not as good of a fit, but you can kind of see that maybe we're getting the start of a hump as you would expect from the response for this species. And then these are just a couple more species that we have run the equations for. Here we have yarrow or Achillea millefolium, and we're seeing that while some of our data matches pretty closely at around 4 to 6 kilograms per hectare per year, we're getting quite a wide range of probability of occurrence. That could probably be to how common the species is across parks. So there's a lot of different, the variation in the other environmental factors might be impacting that. And here with Phleum pratense, we're seeing, again, it's fairly close, but we're have some spread there. So I think what these results show, first, you can really see the limitations of our deposition gradient that we're dealing with, because we're not really seeing what's happening above 7 kilograms per hectare per year. And second, since we're dealing with different spatial distributions of these species, we might need to adjust the models to account for that. So what this study means so far, I think it's on its way to helping provide a framework for assessing individual species in parks, which can be really useful. And as I mentioned before, it allows for a multi-scale view of park response to nitrogen deposition. So we can look at an individual species in an individual park, or we can look at a species across multiple parks, a group of species across parks, or sets of parks dealing or scalar flexibility that larger scale critical loads might not allow. And then it allows for parks to enact more specific environmental management practices. So I think one of the advantages of this could be really identifying location and species specific target loads, which could be really useful. And then I think this also could really help with external stakeholder communication. So being able to specifically mention a species, maybe a threatened species, and how it's dealing with nitrogen deposition, I think that could be a really powerful tool for how we deal with protecting natural resources in national parks. So some next steps. I mentioned a few of these already. The first would be to expand our analysis to include more parks. So moving beyond just our 9 case study parks, because with that, we just have these little pockets of locations for a species. So that could provide, could provide more variation in our nitrogen deposition data. And then second is analyzing the results with a wider deposition gradient. Part of that is expanding the parks included our analysis, but the other part, and something we're working on right now, is redoing this analysis, but replacing the 2016 to 2018 nitrogen and sulfur deposition data that we had originally used for the input, and replacing that with 2000 to 2012. So just trying to expand that gradient a bit to more closely fit the original models. And then something else we're also doing is concurrently is doing the same work, but evaluating it for sulfur critical loads for these same species. So I think so far this analysis shows that there's some good results so far, but I think there's definitely a lot to figure out moving forward. And that's pretty much what I had. I just wanted to also thank Mike Bell and Emmy Felker-Quinn for their help with this research. And also thanks to Alice and Laura for help providing some of the data. And that's what I got.
Thank you very much, Nick. And we have about 5 minutes left. And let's see if we've got any questions in the chat box. I don't think so. Anybody out there? I've got a question. Go ahead and unmute yourself and ask it. We've got a compliment.
Thanks.
Great job, Nick. Well, I'll ask a question. Now that you've gotten a little start on this, like, what kind of questions do you have? What has it made you think about?
That's a good question. I think the main thing I've been thinking about, particularly with this analysis, is thinking about how like the original data from the Clark analysis is more spread out than any of the data we're going to get or from national park, because those are little sort of pockets. So I've been curious about how that will impact our models, trying to apply those models in national parks.
Me not having the fuller data set.
Yeah.
Mhm.
So yeah, this has been my first foray into critical loads, and I've learned a lot.
Great.
Nick, this is Mike. The Phelan figure made it look like there were two kind of distinct relationships occurring there when you had all the points together. Have you looked at the spatial distribution of those to see if it's like spatially relevant? Because you have like the steep points at the beginning, then like a softer slope on the outside.
Yeah. So I have looked at some, I have mapped for some of these species. I don't think I have mapped for phleum yet, but that would definitely be interesting to look at. When I first created that chart, I was wondering what was going on there. So that's definitely something I can look into.
Any other comments or other questions for the previous speakers? We have a couple more minutes.
Hey, Greg, this is Mike. I'll respond to your question in the box asking if there's surveys in parks and other sensitive ecosystems to ground truth and calibrate critical loads. The national park, that's kind of what we're hoping to do with some of these analyses is since the data set is outside of National Park Service plots, is identify what data we have. And initially we were checking to see if it's in the same range of environmental conditions. And then we're hoping to be able to either run a similar analysis separately or include them into a future analysis. One of the problems with most national park data is it doesn't since they are in more pristine areas, we don't have as steep a deposition gradients, especially in the West, to look at species responses, which is part of the reason it hasn't been included in the past. But we're hopeful to have enough of it to calibrate against.
Great. Greg, did you have a quick question? Greg?
That was it. Mike answered it. Thank you.
Okay, good. Well, thank you very much, Nick, for the presentation. Glad you're embarking on critical loads, and hope you'll stay with it. Yeah. Our next speaker is Jason Lynch.
Okay, good afternoon, everyone. I'm Selma Eisel, and as Jason said, this is a tag team between myself and him. I am going to provide a very brief and very rapidly delivered intro, so Jason can have the bulk of the time to tell you about his case study. Next slide, Jason. Okay, so Jason's case study focuses on areas in the Great Smoky National Park above 1,000 meters, but on the East Coast, it was found that typically areas above 800 meters can be frequently impacted by cloud. And it was found from past studies on the East Coast that cloud water samples can be typically 5 to 20 times more acidic than just rainwater. And this type of large loading in such environments is due to a combo of factors such as high frequency of cloud immersion, high wind speeds, orographic enhancement of precip, and large leaf area indices of tree species that are typical of such environments. And not to leave fog deposition out, fog impacted coastal ecosystems also experience higher pollutant loadings very similar to cloud impacted high elevation sites on the East Coast. Next slide, please. In the eastern US, there are 50,000 square kilometers of areas above 800 meters where cloud deposition is an important component of total depth to ecosystems. And the shaded areas on the map to the right denote areas in the East Coast that are above 800 meters. And as you can see in the south, that includes the Great Smoky Mountain National Park and the Shenandoah National Park. And up in the Northeast, we have the Catskills in Southern New York and the Adirondack Mountains in upstate, and Green Mountains of Vermont, White Mountains of New Hampshire, and some areas in Western Maine. And there are also 12 Class 1 and wilderness areas as far as, and 9 national forests. Next slide, Jason. So the bulk of our data on the East Coast comes from 2 studies conducted in the past, the Mountain Cloud Chemistry Project, which ran for 4 years in the late 80s, and then the Mountain Acid Deposition Program, which started in 1993. And MADPRO's main objectives were to develop cloud water measurement systems that could be used in a network-wide environment and to update the cloud water concentration and deposition data that was collected mainly by the Mountain Cloud Chemistry Project. And the map here shows the 3 main stations from that, Whiteface Mountain in the Adirondacks, White Top Mountain in southwestern Virginia, and Clingman's Dome here in the Great Smoky National Park. And Jason's case study focuses on the Clingman's Dome site. Next slide, please. So cloud and fog deposition is generally not considered when evaluating the risk of nitrogen and sulfur deposition. And this is particularly the case for critical load and exceedance analyses. And I've already mentioned that cloud and fog deposition can be a large component, and ignoring it can lead to an underestimation of the risk of higher elevation communities as well as coastal ecosystems to air pollution. Next slide, please. So a quick look at some results from the Clingman's Dome site in the park. Here we are looking at all 3 components of total deposition, dry, wet, and cloud. We have sulfur depth on the left and nitrogen depth on the right. And all of the cloud deposition estimates were produced by Gary Lovett's cloud model. And you can see that both analytes are coming down from 2001 through 2011 with some interannual variation in there. The dotted lines above 2008 indicate that we didn't collect in 2008. And just a very quick look shows you what a large component of the total deposition budget that cloud is compared to dry and wet. And I threw a map of the park here at the bottom. Since the study focuses on Clingman's Dome, I just wanted to pinpoint exactly where the Clingman's Dome was located within the park. Next slide, please. So the lack of current and much cloud and fog deposition data was identified as a need by NADP's Total Deposition Science Committee. And as such, it ended up being a chapter in the TDEP white paper. And I also gave a talk on it in last year's white paper webinar series, which were coordinated by Christy Morris and Mike Bell. And I threw this TDEP total deposition of nitrogen map from 2011 up as the last slide in that talk. If you focus in and about where Clingman's Dome is, that comes out to about 8 kilograms of nitrogen per hectare per year. From the previous slide though, just the cloud water nitrogen deposition value at Clingman's Dome was 7.947. So pretty much just cloud water nitrogen deposition was equivalent to a whole year of dry and wet. And this deposition value for cloud water is only for 4 months. So I have always wondered, gosh, what would these maps look like if we did include cloud and fog deposition? And what's more, what would critical load values look like for these areas? And so I was talking about this at last year's NADP meeting in Boulder to Jason, and he said, we can do that. We can do a case study and we can take the past data from Clingman's Dome and extrapolate it out to current year and see what that is. And so a year later, Jason has done that. And so I will turn it over to him so he can tell you about it. All yours, Jason.
Great, thank you, Selma. Yeah, and so as Selma pointed out, I'll be talking about the case study here. But before I go into the details, I want to talk a little bit about critical loads. Critical loads, of course, are important tools to evaluate the risk of air pollution and the resulting NNS deposition on sensitive ecosystems. And a critical load is a quantitative estimate of exposure of one or more pollutants. And so in this case, we are going to evaluate N and S, and at times evaluate both of them. And it's has significant harm below with a sensitive species in the environment based on our current knowledge. And I went ahead and just put a small diagram showing what we mean by critical loads. On the x-axis, we have deposition, and then on the y, we have some biological response. And the dotted curve is the actual biological response. And then a critical loads can be set for that, and that's where our dotted lines is. And in the case where we have an exceedance where deposition is above that critical load, we would, it suggests that we would have adverse effects. And so today I'm going to really focus on the exceedances of a subset of critical loads that I've looked at. All right, and as Selma points out, we're going to focus on areas above 1,000 meters in Great Smoky Mountain National Park, and that's the area in green here in those regions. We have a pretty simple approach. We're going to evaluate critical loads, 3 different critical loads, and indicators which are publicly available as part of the National Critical Load Database. We're going to use TDEP deposition. We're going to compare wet and dry deposition versus wet plus dry deposition and cloud and fog. We're looking at the 2 intervals, 2009 and 2011, and that was the last year we have data from the Klingman's Dome site, and then looking at what we would have today as a perspective. And of course, the area of interest is areas above 1,000 meters. And so here's just the deposition. Here's total N deposition. And you can see here, cloud and fog represents 7.5, an addition of 7.5 above what we would have for TDAP. And so that gives us a range of basically 14 to 18 kilograms in the earlier period. Deposition has been declining since 2011. And so if we look at what we have today, we just use a basic correlation between the past data to estimate what the value could be today. Obviously, we're not measuring it, and so it is just an estimate, but for nitrogen, that's an additional, almost additional 5 kilograms of nitrogen here, giving us a total from between 10 and 13. In the case of sulfur deposition, in 2009 to 2011, we had an addition of 6.6, giving us a total between 11.6 and 14.4 across the region. In the case of 2016 and 2018, we estimate that there was a substantial drop, about 1 to 1.67 kilograms of sulfur, and having a total of 3.7 to about 4.5 for the areas above 1,000 meters of sulfur. All right, the critical loads we selected were 3. We looked at aquatic acidification. I chose to use the McDonald et al. The 2014 values. There are other values that could have been used for the park. I ended up using the McDonald's mainly because I'm very comfortable with these values and understand them in more detail. They also provide a complete coverage of the park area as well. And so I have a critical load for each reach within the study area. We also looked at closed canopy herbaceous species richness based on Sincom et al. And then lastly, we looked at forest acidification, the work that McNulty et al. Had done in 2013. The indicators for aquatic acidification, here we used a ANC level of 30 as our indicator level. It provides overall protection of aquatic systems. In the case study, there was 562 stream reaches that were analyzed. For species richness, it's an indication of no net loss of species richness. So we're looking at a community metric. We're looking at understory communities only. And then we evaluated the ecoregion 3 and ecoregion 4 critical load for that. So we weren't using the individual plots, but the ecoregion 3 or 4 level value. Lastly, for forest acidification, this is a critical load to protect soil acidification. It's an overall protection of forest health, and it's at a 1 kilometer grid across the study area. We evaluated exceedances. Exceedance here is just simply the deposition of N and S minus the critical load plus some error evaluation. And an example of an area, a margin area that we use for the aquatic critical loads, we use plus or minus a half a kilogram for ours. And so I think I forgot the decimal point there, but a half a kilogram. And then in the case of an exceedance, it's just an area or water body at risk of ecological harm. Okay, what did we end up finding? All right, I have a series of graphs here. I have the years 2009 to 2011, and then the years 2016 to 2018. The top graphs are just TDEP, so wet and dry, and then the bottom graphs are wet plus dry plus the fog and cloud component. All right, we're looking at critical load exceedances. If it's a blue dot on the map, we are below the critical load. If it's a yellow dot, that is we are right at the critical load within that margin of error. And so those are areas where we are within plus or minus a half a kilogram, in this case of sulfur. And then of course, if we're above the critical load, it's an indicated of a red dot. And all the graphs will have the same shading. And what we found here is in terms of the earlier period, if you just focus on the TDEP estimates, we'd have about a 26% exceedance. If we add cloud and fog deposition, that increases to about a 91%. Looking at the current period, we have a substantial reduction in exceedance rates, about 8%. With cloud or fog would be 16. So we would still have a higher rate of exceedance. Looking at forest acidification, again, these are at 1 kilometer blocks using the same scaling here. If you're blue, you're above. If you're red, blue, if you're below. If you're red, you're above. What we find here is in the earlier period, a 91% exceedance rate. With clouds, we have 100. As you can see here with some substantial decreases, if we just focus on TDEP, we'd have a pretty low exceedance rate, but adding in cloud and fog, you can see the exceedance rate expands substantially to 81%. In the case of species richness, we see similar results. With the earlier period, if you do not consider fog and cloud, just the TDEP estimate, you get a total of 0 exceedance or 100% if you include cloud and fog. In the case of the later period, we have a period where we have an exceedance rate of about 52%. But you can see in the yellow, it's a pretty large area which is right at the critical load. All right, just a few closing remarks. It's important to, based on our analysis, cloud and fog is an important component, and it's something that we should definitely consider in our critical load analysis. You can see here when we consider it, exceedance rates expand from 8 to 78%, which is quite large. And so for the Great Smoky Mountain National Park, by considering cloud and fog deposition, we would, without considering it, we would underestimate the ecological risks of air pollution. And this is likely the same case for much of other areas above 800 or 1,000 meters in the eastern US. And then lastly, for critical loads, for some critical loads, deposition is used to determine the critical load itself. And so for certain areas where fog and cloud deposition is important, it's something that we should consider to use as part of our critical load and our critical load evaluation. And then lastly, I just kind of want to challenge the group out there to think about how we can bring together cloud and fog data into TDEP and other deposition products so that it can be better represented in our national models. And I just want to say thank you to everyone. And we're happy to take some questions.
Great. Well, we have about 2 minutes, so thank you both, Selma and Jason, for that talk. And we have a couple of questions, one from Greg and one from Mike Bell. Greg, would you want to just go ahead and say your question so I don't read the entire thing?
Sure. I just was wondering if there are spatial data products that you could merge with the TDEP products, spatial data on fog and cloud precipitation depth, basically, like as a function of total precipitation or as a proportion of total precipitation, or is this really a phenomenon that's very site specific? Like, it's very important for the Smokies, maybe very important for the Pacific Northwest, and maybe not so much for, other places without fog.
Do you want to grab that, Selma?
I would say it's pretty site specific, Greg. It would, yeah. I mean, I think with more data, it could be incorporated into CMAQ possibly. And that was something I had asked Donna about it, if CMAQ addresses it, and she said it currently doesn't. But I know in the Northeast, cloud impaction is spottier. Well, especially if you're hovering around 800 meters. So I'm not sure about the spatial part that you're asking about. I mean, it would be good to investigate it though, but it can be highly site specific.
Yeah, I imagine it'd be a big deal, like for the redwood forests, places that have a lot of fog. So it's cool. I guess the take-home message then, correct me if I'm wrong, is that if you're dealing with an ecosystem that has significant cloud and fog deposition, you would be wise to account for that. Try to account for that if you're trying to do critical loads for those kinds of habitats.
Mm-hmm.
Yes, and you mentioned the redwoods, and it's very important fog for redwoods because they get most of their moisture from fog inundation, especially in the drier season. So.
Okay, cool. Thanks.
Fog is pretty common in the Midwest and along the Gulf Coast too. Certain times of the year.
Sure, there's a lot of it. And Selma and Jason, there's there are actually quite a few questions accumulated in the chat box, which we won't have time to address, but perhaps you could write your answers there for everyone to see, and we'll go on to our next speaker. So thank you both very much. And
Thank
our next speaker, something else? Jason? Okay. Our next speaker is Megan Pettix, and she'll be talking about assessing nitrogen critical loads at North Cascades National Park. And welcome, Megan. We can see your screen, so please proceed.
Perfect, thanks. Can you see that laser pointer?
Yes, we can see it.
Okay, awesome. So thank you all for tuning in for my talk. My name's Megan Pettix, and I'm a PhD student at Washington State University in the School of Biological Sciences. And today I'll be discussing my work focused on assessing nitrogen critical loads at North Cascades National Park Service Complex. And I would first like to acknowledge my co-authors on this presentation, Mike Bell and Tawny Cummings from the National Park Service, and Alita Melsa and Dave Evans from Washington State University. So this audience is well acquainted with the impact that human activity has had on the nitrogen cycle. Transportation, industry, and agriculture all emit reactive nitrogen into the atmosphere. Nitrogen emissions from transportation and industry are dominated by nitrogen oxides, while emissions from agriculture are mainly reduced reactive nitrogen forms, namely ammonia and ammonium. Nitrogen emissions can be transported and deposited back onto terrestrial and aquatic ecosystems as atmospheric deposition. And over time, excess nitrogen deposition alters biodiversity and plant and soil chemistry with cascading effects through ecosystems. Anthropogenic nitrogen deposition has already contributed to ecosystem changes across the Western US, including disruptions in nutrient cycling, ecosystem functioning, and shifts in community composition of certain organisms. Nitrogen critical loads can be developed to assess the threat of nitrogen deposition to ecosystems. A critical load exceedance occurs if ambient deposition is greater than a defined critical load and implies that ecological changes specified by that critical load would be anticipated. So identifying areas where current deposition exceeds critical loads can help air quality regulators and land managers determine where emission reductions are most needed. Accurate measurements of nitrogen deposition are needed for land managers to determine nitrogen critical loads and exceedance estimates. The deposition estimate used to evaluate a critical load can influence whether an exceedance is expected and can change management and policy response. Typically, nitrogen deposition estimates come from ambient monitoring, atmospheric modeling, or a combination of both. In this figure, the map on the left shows National Trends Network monitoring sites where wet deposition is observed. And then the next map shows deposition of total nitrogen estimated by CMAC, which is one of the most commonly used atmospheric models. And then the map all the way on the right shows deposition of total nitrogen estimated by TDEP, which is a fusion of the modeled and observed data. In addition to ambient monitoring and atmospheric modeling, biomonitors such as epiphytic lichens can be used to assess and predict nitrogen deposition. Lichens have long been useful indicators of air quality because they have a high surface area and lack roots. They readily absorb water and lack specialized barriers to prevent vapor loss, and there is a range of air pollution sensitivities across species. Epiphytic lichens, meaning lichens that grow on shrubs and trees, obtain nutrients from the air, precipitation, and substrate runoff. This means that tissue levels of nitrogen are correlated with ambient atmospheric levels of nitrogen. The nitrogen concentration of epiphytic lichens can be utilized to monitor nitrogen deposition in forested areas as a low-cost method to get accurate measurements of nitrogen deposition across a fine or broad spatial scale, even with complex topography. In a study by Root et al. 2013, co-located through-fall nitrogen deposition collectors with nitrogen concentration of collected lichen species. And here you can see the concentration of nitrogen in Platysmacia glauca along the x-axis, and the dissolved inorganic nitrogen in kilograms per hectare per year along the y-axis. And this is showing a regression that they developed, and these dashed lines indicate the confidence intervals and the dotted lines show the prediction intervals. And this regression model effectively estimates through-fall inorganic nitrogen deposition from lichen nitrogen concentration. These nitrogen deposition estimates can then be used to identify areas that are in exceedance of critical loads. Empirical critical loads could be developed based on observations of the response of an ecosystem or ecosystem component, such as lichens, to a given observed deposition level. And assessing nitrogen critical loads based on lichens helps define maximum nitrogen deposition levels that may prevent negative impacts to the broader forest ecosystem. So why is this research necessary? As I discussed earlier, accurate measurements of nitrogen deposition are needed for land managers to determine nitrogen critical loads and critical load exceedances. However, nitrogen deposition estimates from atmospheric models, such as CMAC, can have a high degree of uncertainty, especially in complex terrain. The model data fusion approach, namely TDEP, is reliant on measured values that come from instrumented monitoring sites that are sparsely located throughout the Western US. So there's a need for greater accuracy in nitrogen deposition estimates, especially across the Western US, where there's a vast array of public lands that may contain ecosystems that are sensitive to elevated nitrogen deposition. So based on these gaps in knowledge, our research is focused on increasing our understanding of patterns of atmospheric nitrogen deposition in the North Cascades National Park Service Complex, determining the extent to which ecosystems in the park are being affected by nitrogen deposition, and gaining insights into predominant sources of nitrogen emissions affecting the park. So to orient you with our study area, North Cascades National Park is in the northwestern corner of Washington, just below the border with Canada, and I may refer to it by its abbreviation NOCA from now on in this talk. So it's a park that's known for its jagged peaks crowned by more than 300 glaciers. It spans the Cascade Crest from the temperate rainforest of the wet west side of the state to the dry ponderosa pine ecosystem of the east. And it consists of several units, including the North and South unit of North Cascades National Park, Ross Lake National Recreation Area, and Lake Chelan National Recreation Area. And I established lichen air quality plots across NOCA. I identified 10 distinct spatial areas across 3 transects within NOCA that represent both north to south and west to east gradients within the park. And at each one of these spatial areas, I established 3 plots along an elevational gradient from low to mid to high elevation for a total of 30 plots across the park. The lichen air quality plots were 114-foot radius circular plots with an area of 0.9 acres. And at each plot, I collected lichen tissue samples for elemental analysis according to US Forest Service protocols. And here are a few photos of me collecting lichen tissue samples in the North Cascades. And after that, I brought the samples back to the WSU Stable Isotope Core facility and worked on processing them and analyzing them for both nitrogen content and nitrogen stable isotope composition. So for an initial interpretation of the lichen nitrogen content from my collected samples, I compared my values with the provisional element analysis thresholds for percent nitrogen for my focal species determined by the US Forest Service. And values above these concentrations are considered elevated, and values at or below these concentrations are considered within background range. And down here you can see the provisional nitrogen threshold for Platysmacia glauca, and that's 0.59% nitrogen. So a nitrogen content above this value would indicate elevated nitrogen deposition, and a value below would indicate background levels of nitrogen deposition. So this figure is showing lichen nitrogen percent data for 2 of my focal lichen species, Platysmacia glauca in gray and Alectoria sarmentosa in green. Lichen nitrogen percent is along the y-axis, and the longitude of the air quality plots in decimal degrees is along the x-axis, going from west to east across the park. And I added these dashed lines to show the value of the provisional thresholds for both of the species, 0.59 for Platysmacia glauca and 0.65% nitrogen for Alectoria sarmentosa. We see that all of these points fall below the respective thresholds, indicating that these plot locations likely have low background levels of nitrogen deposition. However, we do see some points with values upwards towards the thresholds, indicating that there may be areas with slightly higher nitrogen deposition. Looking at this data spatially, just focusing on one of the collected lichen species, Platysmacia glauca, we can see which areas of the park have the highest percent nitrogen values, suggesting higher rates of nitrogen deposition. The color ramp shown here goes from dark blue to red, going from low to high percent nitrogen values. And we can see this area here in the southwestern region of the park, which is the Cascade Pass area, has some of the highest percent nitrogen values for Platysmacia glauca, upward to 0.5% nitrogen, which is just slightly below the provisional threshold for this species. So I'm now going to show this same map displaying percent nitrogen values for Platysmacia glauca overlain over the National Land Cover Database layer, zoomed out to show the regions surrounding the North Cascades. And here are the land cover classifications. So we can see the areas immediately surrounding the North Cascades are mostly forested, but in close proximity, there's acreage that's light yellow, which indicates pasture for hay and livestock. There's acreage that's dark brown, indicating cultivated crops, and acreage that's varying shades of red, with light red being low intensity development and deep red being high intensity development. The western edge of the park is in closer proximity to these human impacted land cover classifications. And this includes that Cascade Pass area that I pointed out here that had the highest percent nitrogen values. And it's situated closest to this large red shaded area here, which is the Seattle metropolitan area. So proximity to nitrogen pollution sources, as well as landscape features and meteorological variables, are likely influencing rates of nitrogen deposition in the park. And analyzing lichen nitrogen content will help us assess patterns of nitrogen deposition across the North Cascades. I will also be assessing the nitrogen stable isotope composition of collected lichen samples to assess dominant sources of nitrogen pollution. So as I mentioned earlier, lichens are totally reliant on atmospheric sources of nutrition. And not only can lichen nitrogen tissue content be correlated with ambient levels of atmospheric nitrogen, but lichen nitrogen stable isotope composition can be used as an integrated measure of the nitrogen stable isotope composition of atmospheric deposition. And this concept comes from foundational work done over in Europe in amphitrophic bogs, which are peat accumulating ecosystems that are fed only by atmospheric deposition. And this figure here is from Bragaza et al. 2005, and it's showing the delta 15 nitrogen of sphagnum peat moss along the y-axis, and along the x-axis is the NHX to NOx ratio in atmospheric deposition, which is the ratio of reduced to oxidized nitrogen compounds in both dry and wet deposition. And the different symbols are the different study sites where moss was collected, located across 11 different European countries, which you can see here. Sphagnum plants had delta 15 nitrogen signatures ranging from about -8 to about -3 per mil. And they found that isotopic signatures were related to this ratio of reduced to oxidized nitrogen forms. And this was significant, and the difference was not significant with just the total amount of nitrogen deposition. They found that bogs located in areas characterized by greater emissions of ammonia, which, if you remember from the beginning of this talk, are mainly affected by agricultural activities, had sphagnum moss with lower delta 15 nitrogen compared with bogs that were in areas dominated by nitrogen oxides, which are mainly derived from industrial activities. So their findings suggest that the type of nitrogen prevailing at a given locality as a consequence of human activities significantly influences the delta 15 nitrogen signature of the mosses. So looking at my data collected from the North Cascades, focusing again on just one of the collected lichen species, Platysmacia glauca, we see that the delta 15 N values were very negative, ranging from -6.9 to -4.1 per mil. The lowest values are in dark blue, then light blue, yellow, and red indicates the most positive values. We see that delta 15 nitrogen tended to be more positive with increasing elevation, which you could really see here in the northwestern corner of the park, where these 3 transects are going from high to low elevation for those 3 sites. And we also see certain areas of the park that have more positive values, including this Cascade Pass area that I highlighted earlier. And if you recall from the land cover map I just showed, this region of the park is in the closest proximity to the Seattle metropolitan area. And with more positive delta 15 nitrogen signatures, this suggests that oxidized forms of nitrogen derived from industry and vehicle exhaust may be a significant component of atmospheric deposition in this area. And for other areas of the park with more negative delta 15 nitrogen signatures, this suggests that reduced forms of nitrogen derived from agricultural sources may be a larger component of atmospheric nitrogen deposition in these regions. So the next step for this project is to integrate lichen data and atmospheric chemistry models in a GIS framework to develop a powerful approach to evaluate nitrogen critical load exceedance across the Pacific Northwest. I'll not only be incorporating the lichen data that I collected from the North Cascades, but will also be incorporated throughout Region 6, which encompasses the states of Washington and Oregon. And once we have developed a methodology, we're hopefully going to be able to assess patterns of nitrogen deposition across the Pacific North region and assess the factors that are influencing the observed patterns of nitrogen deposition. Now, as I mentioned earlier, previous analyses have shown that modeled nitrogen deposition has a high degree of uncertainty in mountainous regions. We expect greater accuracy of nitrogen deposition estimates in these regions due to the greater spatial distribution and density of air quality sampling with this integration with lichen data. Regional lichen nitrogen stable isotope composition will help us gain insights into emission chemistries and predominant sources of nitrogen emissions impacting the region. And then additional explanatory variables will include precipitation, elevation, and a variety of other landscape features. Have this study be a template for assessing nitrogen deposition and nitrogen critical loads of mountainous regions throughout the US. With that, I would like to acknowledge a lot of my collaborators, the WSU School of Biological Sciences, and collaborators with the National Park Service Air Resources Division, the US Forest Service, and my graduate committee. And last but not least, my field crews for 2018 and 2019 who helped me collect all this data. And if there's time, I will take any questions. And if I ran over, I listed my email address here where you can follow up with me if you would like.
No, you stayed within your time. Thanks, Megan. We cheated you about a minute and a half, so we were a little bit late starting. So let's take a few minutes for questions. That was, of course, I would think that it was a pretty interesting talk.
Thank you.
I try unsharing so we can see folks and see if we've got some questions. Great.
I see one question about why did I choose those 2 species and how the results differed between the 2. So I really focused on results primarily today from one of the lichen species, Platysmacia glauca, and I utilized that because I wanted to make sure that my data I collected in the North Cascades was comparable with the very extensive data set that the US Forest Service has. And they have a short list of different lichen species that they've selected based on physiological characteristics of those species and their different sensitivities. Platysmacia glauca was pretty ubiquitous across the park, but I did have to add in a few other species because it wasn't actually present in all the different areas I wanted to sample. I mentioned that the North Cascade spans the west to the east side of the Cascade crest, so I did have to incorporate 2 other lichen species, Lotharia vulpina and Lotharia columbiana, that were present over on the east side in that drier ecosystem. So I guess to answer your question, it was kind of a matter of what lichens were available and the US Forest Service data set. And the results differed in terms of their percent nitrogen. You kind of have to calibrate it by each species because they have, you know, different physiologies and kind of absorb nitrogen in different ways. So I saw similar patterns for the different species once they were calibrated. And for the nitrogen stable isotope data, that should be pretty standard, where you can interpret those values across the different species quite equally. And I did find that my values were consistent between species.
Great. Well, thank you again, Megan, for a very nice talk. And if you have questions for her, go ahead and write them in the chat box, and we'll get ready for our next speaker and our final talk of the session with Sarah Anderson and co-authors. Okay.
All right. Hopefully my title slide is coming through and sound is okay today.
Sounds fine. We can see your slide.
All right, great. Good afternoon, everyone. Glad to be wrapping up the session, and it's been a great session looking at critical loads and all of our different applications and recent work with them. It's exciting to see it all. And today what I'd like to talk to you all about and kind of introduce you the CLAD and NADP community to is the Resources Planning Act, and what we're doing with the Resources Planning Act to begin to incorporate air quality into this assessment that the Forest Service does every 10 years by using critical loads and atmospheric deposition, and working with Claire O'D, who many of you know, as well as Jen Costanza, all of us at the Forest Service. And so kind of a roadmap for this talk, we're gonna go through it all. We're gonna start at the past and introduce you all to the Resources Planning Act and the assessments that have stemmed from this act that is a requirement for the Forest Service to do every 10 years. And then want to share with you what we're doing to begin to incorporate air quality more explicitly into the Resources Planning Act assessment, or RPA assessment. And then finally, want to share with you our vision for the future. We really see this as a really unique platform to look at the impacts of air quality on natural resources and renewable resources, as well as to think about what those impacts might be into the future and do the work to project those impacts into the future. And by combining deposition and critical loads, I think we have a very strong framework for ways to look at what those impacts could be under different scenarios. And so to begin, introducing you to the RPA assessment. It was established by the Forest and Rangeland Renewable Resources Planning Act of 1974. It mandates that we at the Forest Service do a national report every 10 years on the conditions and trends of renewable resources across all forest and rangeland ecosystems or systems. So that includes both those on Forest Service land and off Forest Service land. So it is, you know, a coast to coast assessment for US lands. The goals of the assessment are every 10 years to provide, namely, a snapshot of current conditions on our forest and rangelands to understand their current condition as well as trends, and identify the things and various drivers and stressors affecting those lands and the resources that they do have, and then to take that and project it 50 years into the future, and look through the various scenarios of what might be happening and what might need to be accounted for. And so for this version of the RPA assessment, our projections will span from 2020 to 2070. And what we focus on in the RPA assessment is the interaction of economic, social, and biophysical factors that, coming out of the language of the Act, affecting the productivity of our forest and rangelands ecosystems. And so really looking at that intersection between all of those different things to better understand the resources that we do have on the landscape. And so to do those projections into the future, the RPA has developed a set of scenarios to kind of get a feel for the range of different possible futures. And so what the scenarios incorporate are two different elements, both coming from the IPCC. First, incorporating climate into our future scenarios using the representative concentration pathways, RCPs, 4.5 and 8.5, to capture that climate and biophysical element. And then on the other hand, incorporating into the RPA scenarios, 4 of the different shared socioeconomic pathways also coming from the IPCC, and together integrating both the climate and the socioeconomic elements into kind of core scenarios for the RPA. And this is what those 4 core scenarios end up looking like. Along the x-axis, you can see we have the emissions scenarios, the RCP 4.5 being our low emissions future versus our high emissions future using the 8.5, RCP 8.5. And then you can see we have a range of different socioeconomic futures capturing socioeconomic growth ranging from low to medium to high. And so by integrating the climate and the socioeconomic together, we have these core scenarios that are used by all resource areas to evaluate and to project future conditions going forward. And so what resources do we look at? Well, the RPA looks at a lot. Here's kind of a list that I'll tally up here. Really focused on looking at both land resources, what is on the landscape, how land cover, land use, as well as forest resources, forest types, urban forests in particular. New in this cycle of the Resources Planning Act assessment is a forest or a disturbance chapter. That actually deals with forest and rangeland, typo there. But then we have forest products, so everything from pulp and paper to saw logs and timber, and as well as the carbon dynamics, getting especially at that climate element, and then rangeland resources, especially grazing, as well as water resources, wildlife, fish, and biodiversity, and outdoor recreation make up the core chapters within the RPA assessment. And so initially, the RPA assessment was very focused on the kind of the economic concerns related to renewable resources, especially the supply and demand of our forest products. But over the years, the emphasis has really broadened from that initial element of focus to really have a much more holistic picture and concern about our resource conditions, as well as ecosystem health and sustainability. And what this broader scope allows for this broader scope is very much permitted right in the Act. The Act specifically states that the assessment that we do every year is not limited to the requirements listed there, but rather to be a bit more flexible in order to capture all of the factors that affect our natural resources and our renewable resources that we have in our forested and rangeland ecosystems. And so, as I think we all know here, and we've all seen, air quality and atmospheric deposition does impact those resources. And it impacts our forested resources, our water resources, our herbaceous plants and plant communities across the landscape, and our lichens. Linda, I had to throw that picture in for you. I Googled atmospheric deposition. It was the 3rd one that came up, literally with forests, waters, and then lichens. So I think you're having an effect on the world. And so we all know that we have all sorts of impacts across, and we've heard about them today. We hear about them at NADP all the time. And so knowing that, what our goal has been is to really introduce, make more explicit this idea of air quality in the RPA assessment. And the way we're doing this is through a sidebar in the disturbance chapter of the RPA, which I referenced before. It's a new chapter in this cycle of the RPA assessment, and we're excited to have it in there. And we're also excited to have this, although it's short, it's just a sidebar, just a couple of pages, to begin to introduce this concept more explicitly. Prior versions of the RPA have very shallowly referenced air quality as an influencing factor, and deposition has come up in some of the modeling with the carbon dynamics that the RPA assessment looks like at. The forest resources chapter of the RPA has a watershed health analysis that they do that takes into account acidification, but it's these air quality ties are buried very deeply within the RPA. And what we want to do is we want to pull it out and be able to talk about it more explicitly. And so what we see this sidebar doing is providing a starting point for future work and expanding the scope of the work that we can do to analyze and project air quality impacts on, broadly speaking, but, specifically on our renewable resources as the Act calls for. And then the other thing is to help build some of these connections between the air quality impacts as well and the different resource areas that the RPA addresses. And so we have to give a huge shout out and tons of thanks, which is also another reason for this talk, to the CLAD group for creating the Critical Loads Mapper tool. Is the main resource that we're using within this air quality sidebar. And what we've done is, besides introducing air quality and the scope of the different types of impacts that it can have, is really use the Critical Loads Mapper to highlight deposition and critical loads, as well as critical load exceedances, to begin to provide a short case study of how this can work and how this impacts our natural resources. And so using the Critical Load Mapper tool to extract atmospheric deposition data, as well as using it for the critical loads. Although we do talk about terrestrial and aquatic ecosystems, due to our tight length constraints, we've really focused in on the surface water acidification critical loads. This mostly has to do with the fact that the water resources chapter is one of the few places in the RPA where we already have that tie to deposition and air quality more broadly. And so wanting to leverage that as we try to open this door, or open this window to hopefully a bigger conversation going forward. And then using the deposition along with the critical load to look at the mapped critical load exceedances, of how to look at the trends that have gotten us here historically, our current condition on the landscape, as well as how we might be able to use that to project into the future. And so, for reference, these are our 4 RPA regions. They are divided into subregions, although for this analysis and to keep things as simple as possible, we use the larger 4 regions here. And so we took the deposition data from the Critical Loads Mapper tool, summarized it by the RPA regions you see here, to begin to look at those trends over time and really pull out begin to highlight the differences as you move from one part of the country to the other at a very broad brush stroke. And so using the CMIP5 deposition models that are available in the Critical Loads Mapper tool, a little bit of information of those coming from the community atmospheric model based on the historical emissions in retro and the Edgar Hyde, as well as then using those future projections that are already future projected deposition from the Critical Load Mapper tool, focusing in on the representative concentration pathways 4.5 and 8.5 to match the scenarios that the RPA assessment is already using. And this deposition story is one that I think is familiar to many of us. Historically, deposition was pretty low, pretty much across the whole country. And then you can see in our different RPA regions, and really all of them, the increases over time, peaking in the late 20th century, and then decreases, although the differences between the two different and also what we were able to do with that is, since we know the deposition and we have critical loads, specifically we use the critical load for surface water acidification. I should point out that this is deposition, nitrogen and sulfur, total acid deposition in equivalence. And so then what we were able to do with the deposition and having the critical loads is start looking at where those exceedances are. And so starting off with, we have kind of 4 maps to highlight key points in that deposition story over time, starting with the historical deposition at our earliest time point in 8 pier, a lot, especially in the east, of right around when emissions and deposition were peaking, this one specifically from 1980, and then using, although it is projected, kind of current map 20, and then into the future, the further decrease in critical load exceedance is expected, even under this map is for representative concentration pathway 8.5. So you know, we have this great story to tell in this short sidebar, and then we end the story with trying to build some of these connections to many other RPA resource areas. And as you know, we heard in the talks today, starting from in this entire session, there's, you know, critical loads relevant to so many of the different resource areas that the RPA covers. You know, the biodiversity trees and herbaceous plants. That's a key portion of Chapter 10 in the RPA assessment. Watershed health. You know, we've Jason touched on some of the water acidification stuff, or water critical loads. Water resources is Chapter 9 in the RPA assessment. Growth and survivorship of trees. Thank you, Emmy, for giving us that presentation earlier today, directly related to Chapter 6 of the forest resources chapter in the RPA assessment. And then we know about there's interactions between, you know, invasive plants being, you know, increased growth under increased nitrogen deposition, as well as interactions with fire. And these interplays between these different disturbances with both invasive species and fire being key disturbances. These aren't the only connections between next iteration of the RPA assessment. And so thank you very much for your time. If you'd like to learn more about the RPA assessment, it is not just a report, it is a lot of analysis, a lot of different data sets are created by the RPA scientist team. I encourage you to contact Claire O'D, who is currently the assessment program coordinator. I think that's her. I think I got that title right. And she is a great resource for providing connections to the folks doing a lot of the great analyses, including the projections into the future and for all of our resource areas. And so with that, I think there should be a little bit of time for questions if there are any.
Thank you, Sarah. Thanks not only for the information about you know, applicable loads in this context, but also for introducing the RPA and all the great information that it covers. So yes, we do have a couple minutes, and if you'll switch to the non-share mode, we'll see what questions are out there. Looks like we've got at least one in the chat. Oh, from your co-author, Claire.
Yeah, the RPA is just truly, I got to work with the team for 4 months on a detail, and is truly a great group of folks doing really incredible work across, you know, the whole spectrum. So.
Hey Sarah, it's Mike.
Hey, Mike.
Cool to see so many direct uses of the critical load mapper tool, and I know you brought up some of the needs when we were talking about this at the spring meeting. But like, if there are any types of data needs or like actual direct uses that aren't currently available, or data sets that would be of value to have in there, like outside of the obvious tree, like in main critical load things, just, yeah, please let us know as we're trying to prioritize and figure out what to put in there. And if there's something that we know is going to be used, it should rise to the top.
Yeah, I know, speaking from, if I kind of can split between my RPA hat and my forest management hat, having more critical loads related to our rangeland resources would be incredibly valuable. Thinking about, you know, the grassland systems, as well as some of the other more non-forested systems out there. We seem to get really tied to trees, which is, you know, important. I shouldn't say anything. I'm, you know, employed by the Forest Service, but the other side of things is that 1/3 of the land we manage is non-forested systems, and so figuring out ways to better capture those, I think that would also be really beneficial to the RPA because of the focus on forest and rangeland systems. And it's one element where I could see, you know, a lot of room for improvement. There's enough there to get us something, but I can see a lot of potential there.
Okay, thanks.
Yeah, I also, one other thought is just thinking about some of the scientists that we're working with on the RPA. There could be a lot of potential for more specific needs, you know, especially related to the biodiversity stuff. There could be a lot of potential there that I can think of. Granted, I'm not doing the analysis, so I can come up with lots of ideas and hopefully pass them to someone else.
Well, that brings us to the end of our session. And are there any other questions? And we don't have anybody behind us, so we could hang on for a few more minutes. There's somebody else that would like to talk, but Greg, did you have a question?
Well, I just wanted to congratulate you on a great session, and thank you, Linda, for moderating the session and being the chair of the session, and some really cool talks. So Krish is just finishing up his presentation. He's not quite to the conclusions yet on link 1, but getting there. Oh, here they are. So I could just show them to you. So I don't know if this will work, but
there we go. Our concurrent session.
Yeah, concurrent session. You probably can't even read that. So anyway, sorry, thought it might work, but it doesn't. So I want to remind everybody that tomorrow morning we'll be starting about 9 a.m. for atmospheric deposition and changing society. And then the poster session following that, there's a bunch of critical loads posters and related posters. And so please visit that. I'm going to sign out now and go say the same thing to the other session, but that session has been recorded and you'll be able to view that later. By my assessment, not being a critical loads expert, I think that Krish's talk is probably the one that has the most cross-pollination. So that's probably one you'd want to see. And again, tomorrow morning, 9 a.m. Eastern, we'll have the last session. Thanks so much, everybody. And you guys are welcome to hang out here. I know CLAD is a social group and you're a model of how scientists should interact after hours. And so, you know, please hang out, I guess, if you like. I guess David will kick you out when the time comes. So thanks once again.
Thank you, Greg. So, Mike, did we have a social hour scheduled?
We had one Tuesday. We didn't have one for today.
Oh, okay.
It's in the fridge.
Okay, great.
I like getting permission. When someone gives me permission to open a beer at 2 o'clock during the day. So.
Well, it's after 4 here.
Yeah.
Okay.
Are you back east now?
Yes. Mm-hmm.
That's too bad.
She came back to our dark side.
Pretty nice here.
The East is so bad, Mike.
I have unqualified opinions. But no, every time I go, I like it more, and I'm not there enough to have changed that core belief that it's not as good as the
that will convert you. I was really jealous when Jim Renfro was talking about the peak leaves on Tuesday. That just was beautiful.
Yeah.
Yeah. Summers ago to like, it gave me a new appreciation. But yeah, just like the diversity of life that was there was, I don't know, I guess unexpected.
Species. I mean, that's like amazing.
Yeah, that's just it.
Plus, Chris is slacking.
I definitely agree on the biodiversity theme for the East. And that does include insects much more dangerous here. And birds, it's really nice. Oh, well, I feel like we're holding everyone up. But everybody, you're welcome off if you like, or just stay on and chat. Thank you, everybody, for joining us today.
Chris did just there's someone has a real question before I.
And we are right on schedule with the 10-year decadal process.